Okay, thank you. Appreciate that one. All right, so let's see. So last time we went over the first part of the PowerPoint B of chapter 3. We compare and contrast the cell membrane of the prokaryotes versus the cell membrane of the eukaryotes, which is essentially the same, except that the prokaryotic cell membrane does not have cholesterol, whereas the cell membrane of eukaryotes has the phospholipids, has the proteins, complex carbohydrates, and cholesterol. Remember, however, that among the prokaryotes, we did have one exception, which was bacteria in the genus Mycoplasma, which do not have a cell wall, therefore they do have sterols in their cell membrane. We talked about the function of the cell membrane in prokaryotes, which is essentially the same as the function of the cell membrane in eukaryotes, except that in prokaryotes, the cell membrane is also the site of ATP synthesis, which in eukaryotes, the place where ATP is made is the mitochondria. We talked about diffusion and how substances diffuse across the cell membrane. And we said that cells that are immersed in a hypertonic environment, hypertonic to the inside of the cell, will lose water to the environment and will shrink, whereas cells immersed in a hypotonic environment, which is hypotonic to the inside of the cell, the cells will take on water and begin to swell and eventually may burst, may explode. And we said bacterial cells are adapted to hypotonic environments because the cell wall of bacteria is going to prevent the bursting of the cell in a hypotonic environment. So the cell takes on water, the cell membrane swells, but the rigid cell wall will prevent the cell membrane from bursting. So the bacterial cells are adapted to hypotonic environments. The cell wall is protecting them from hypotonic environments. So if you want to harm cells, you need to submerge them or expose them to a hypertonic environment where the cell is going to lose water, dry out, and die. So examples of hypertonic environments that are used to preserve foods are doing that, like salted fish and dried meats and jellies with lots of sugar. All those are hypertonic environments. And those kinds of foods last for a long time because bacteria cannot colonize them. They shrink, they dry out when they try to colonize those foods. We talked a little bit about, what did we talk about? Yeah, we talked about these already. Other forms of transport like active transport and facilitated transport. Yeah, a facilitated transport is a protein that transports substances from one side of the cell to the other using simple diffusion. Whereas active transport, and the key with facilitated transport is that there is no energy required. The difference between active transport and facilitated diffusion is that active transport requires energy. Okay. All right, active transport requires energy in the form of ATP or sometimes ions like hydrogen ions to move substances against a concentration gradient. So diffusion requires a concentration gradient, active transport does not. And this should not be new information for you guys. You should have seen this in Biology 426. So the other thing we said was that lipids, the phospholipids that make up the cell membrane in prokaryotes are similar enough to the phospholipids that make up the cell membrane in eukaryotes, where ultimately cell membranes are not a good target for antibiotics because they don't have, there isn't enough differences between prokaryotic cell membranes and eukaryotic cell membranes. So, okay, so now cell wall. We're going to spend a little bit of time on the cell wall. This is a very important topic. Remember that the function of a cell wall in bacteria is to prevent lysis in hypotonic environments. We just got through reviewing that principle. The cell wall surrounds the cell on the outside of the cell membrane. A medical importance of cell wall, this is the action of some antibiotics, of some of the best antibiotics, because eukaryotes do not, especially animals, do not have a cell wall. So any antibiotic that targets the cell wall of bacteria will be harmless to humans because we don't have a cell wall. The cell wall can make organisms more virulent, more pathogenic, and we can use them to identify organisms. We talked already about that, about how we can classify bacteria based on whether they have a gram-positive or a gram-negative cell wall. We mentioned from day one that the cell wall of bacteria is made of a substance called peptidoglycan. Murine is another term for peptidoglycan. This substance is unique to bacteria and essentially consists of a disaccharide backbone made by two monosaccharides, N-acetylglucosamine and N-acetylmuramic acid. Both of these are derived from glucose, so they are modified glucoses. So in peptidoglycan, these monomers of glucose, of modified glucose, alternate so that you would have an NAG bound to an NAM, bound to an NAG, bound to an NAM. So you get hopefully the idea. NAG, NAM, NAG, NAM. They're going to alternate, creating these very long chains that are going to surround the cell. Coming from the NAM, there will be a series of 4 amino acids, which they are also unique to bacteria. They are modifications from common amino acids. And through those amino acids, this chain of peptidoglycan, of monosaccharides, disaccharide chain, links to another disaccharide chain. So every NAM has these 4 amino acids that are used to link to another chain. So all the chains are linked together. They're not separated. They're linked together by these cross bridges of amino acids. Okay, so let's get a better picture of these. Here's glucose. This is NAM, and NAG, and NAM, which are modified by from glucose. All right, so these are the tetrapeptide, the dipeptide, the disaccharide chains here, and you can see from the NAMs how we have 4 amino acids that are coming down. Here's another NAM, 4 amino acids coming down, and then those amino acids are linked to each other by what's called a cross bridge. So these connecting bridge right there and right there, that's called a cross bridge. Okay, so you can see how all of these chains of the disaccharide are going to be held together via these cross bridges connecting the tetrapeptide chains. So that is the structure of sorry, just have a, yeah, that is the structure of peptidoglycan. Okay, so definitely know these. Now remember that gram-positive and gram-negative cell walls were slightly different. We're going to look at the gram-positive cell wall first. And gram-positive cell walls have a thick layer of peptidoglycan. And that is essentially what the cell wall, the main component of the cell wall. That's basically almost all there is. So here's the thick layer of peptidoglycan of the gram-positive cell wall. Between the peptidoglycan and the cell membrane, the phospholipid cell membrane, there is a narrow periplasmic space. And anchoring the peptidoglycan cell wall to the cell membrane are these lipoteichoic acids that again help to anchor cell wall to cell membrane. There are also teichoic acids sticking out of the peptidoglycan chains. So teichoic acids and lipoteichoic acids are part of the gram-positive cell wall. So the gram-positive cell wall has a thick layer of peptidoglycan, teichoic acids, lipoteichoic acids. Lipoteichoic acids anchor the cell wall to the cell membrane, and there is a narrow periplasmic space. Okay. So these are other depictions of the cell wall. Because there are no lipids in the cell wall of the gram-positive, when during the decolorization step in the gram staining, the initial stain, the crystal violet, is going to be retained by the cell when it's decolorized, and the cell will retain that purple color. It will not be decolorized, will not lose the first color, and retains the first stain. There are a couple of modifications to this gram-positive cell wall structure. Members of the genus Streptococcus have polysaccharides, complex polysaccharides, sticking out of their gram-positive cell wall. These polysaccharides are complex polysaccharides, so they are antigenic, which creates a source of antigens that can be used to identify the cells. So streptococcal species, a species of the genus Streptococcus, can be identified based on these polysaccharides, which are actually complex polysaccharides because they're stuck to the peptidoglycan. These polysaccharides are unique to each species, so they could be classified based on those polysaccharides via an antigen-antibody reaction. And that's how streptococcal are classified in a large manner, is through those polysaccharides. So that's a modification of the gram-positive cell wall. Another modification of the gram-positive cell wall are the members of the genus Mycobacterium. From chapter 2, we said that Mycobacterium is the bacteria that has the waxy cell wall. So what happens to these cells is that they have the peptidoglycan, and then outside of the peptidoglycan, they begin to build these mycolic acid waxy cell wall. So we have, this is the cell membrane right here. This is cell membrane. They do have peptidoglycan. So classification-wise, these cells are classified as gram-positive. And then outside of the peptidoglycan is the mycolic acid. So this is the wax. Okay. Young cells have a very thin layer of wax. And as the cell ages, the layer of wax becomes thicker and thicker and thicker. So these cells grow very, very slowly because the wax doesn't allow nutrients to go through. So that the older the cell, the thicker the waxy cell wall. So this is again a modification of a gram-positive cell wall. The genus is classified as gram-positive, but it's a special gram-positive. It's an acid-fast gram-positive. Okay, so those are two modifications of gram-positives. Now, let me know if you have a question, and we can address any questions that you may have at any time. All right, the gram-negative cell wall is very, very different. First of all, it does have peptidoglycan, so it will have NAMs, NAGs, and the tetrapeptide chains. However, the layer of peptidoglycan is very thin. There are no teichoic or lipoteichoic acids present, and instead of having a narrow periplasmic space, gram-negative cell walls have a wide periplasmic space. Those are some of the differences, not all the differences, but some of the differences between gram-positives and gram-negatives. Another difference is that the gram-negative cell wall has, outside of the peptidoglycan, another layer of something called the outer membrane. Now, this is a little bit confusing, the fact that a component of a cell wall is being called a membrane. So do not let the name confuse you. This outer membrane is not the cell membrane. This is part of the cell wall. The outer membrane consists in turn of lipopolysaccharides, phospholipids, porins, and lipoproteins. So we'll talk about all these components here in a minute. Let me show you a picture of the gram-negative cell wall. Okay, this right here is the cell membrane. That's the phospholipid cell membrane or plasma membrane. That's not cell wall. This entire area right here is the periplasmic space, and that's why it's going to be called a wide periplasmic space. This thin layer right here, this thin layer right there is the peptidoglycan layer. Okay, so you can see how it's very, very thin. Now, this is the outer membrane, all of these right here. Notice that in the list of components of the outer membrane, we said phospholipids. Okay, phospholipids. Phospholipids are the same molecule that makes up the cell membrane. Here's one layer, one leaf. The cell membrane has two leaves of phospholipid. The outer membrane has one leaf of phospholipids. Okay, so the inner leaf is a phospholipid leaf on this LPS. Okay, so there's the phospholipid. Again, we're going over the layers of the outer membrane. This is the inner leaf. The outer leaf, right here, is the lipopolysaccharide. So it looks like this. There's the little fatty acids, and then it has these things up here. Okay, so this little brown square with the little tail inside and a little hair-looking thing on the outside. This is a lipopolysaccharide. Lipopolysaccharide, lipo means lipid, and polysaccharide is a polysaccharide, it's a sugar, or a carbohydrate, I should say, not a sugar, but a carbohydrate. Okay, so the lipid of the molecule are these two tails right here. These are two fatty acids right there, okay, which they are on the inside facing the fatty acids of the phospholipids because they're fats, and the whole environment is a water environment, so the fats try to stay away from the water. Then we have the little head with a phosphate, and the polysaccharide is what's sticking out. So we have the outer leaflet of the outer membrane is the LPS, which, by the way, LPS is unique to bacteria. Just like the peptidoglycan is unique to bacteria, LPS is also. We talked about the phospholipid inner leaflet. Okay. All right. So that's the LPS and the phospholipid. So the lipopolysaccharides, the LPS, are very important in the cell wall of the gram-negative because it's a great component of the cell wall. Then you have all these proteins embedded here. These proteins right here are especially important. They're called porins, and porins sounds like pores. These are pores. These are gates that allow substances to go through. Keep in mind, when you have two lipid layers like these, it's again very difficult for nutrients and other molecules to make it all the way from out here, cross this layer, cross the peptidoglycan, cross the phospholipid before it makes it into the cytoplasm. So that is very difficult. So to help nutrients go through all these layers, these porine proteins are that. Um, does that work similar to aquaporins? Yes. So some of these porins will be aquaporins. So will be pores that allow for water in. So there are many different types of porins, and each porins or these different types of porins will allow different substances through. So porins that may allow glucose through, or porins that will allow certain amino acids through, or porins that will allow water through. So they're very specific as to what they allow, which means that for a substance to go into a gram-negative cell, it will need a porin. Unless the substance happens to be a fat, which can go through the membrane itself, the substance will need a porin to go through. Otherwise, it will not be able to make it through. So that is one of the drawbacks of trying to find drugs that work with gram-negatives. Is the fact that gram-negatives have the porins, or that the gram-negatives do not allow substances through very easily, so you will need a porin that will accept the antibiotic, for example. Okay, so porins are an important component. The other component are these brown lipoproteins right here, which are going to anchor the outer membrane to the peptidoglycan. So the two are glued together by these lipoproteins. So again, the components of the outer membrane. LPS is one of the major components. And then we have the phospholipid inner leaflet, the porine proteins, and the lipoproteins. So yes, you should know the components of the outer membrane of the gram-negative cell wall. Another thing to remember is that the polysaccharide that sticks out of the lipopolysaccharide is an antigen because it is bound to another molecule. So it's a complex polysaccharide. So it is antigenic. All right. So the lipopolysaccharide is going to have two components. The lipid, which are the two fatty acid molecules on the inside, right there, and the polysaccharide, which is this right here. The lipid, called lipid A. So these two lipids are lipid A. And these lipids are toxic to humans. This is what we call an endotoxin, because it is inside the bacterial cell, and it will not be released until the bacterial cell dies. So as long as the cell wall of the bacteria is intact, this little toxin will not harm us because it's not going to be released. The minute the cell begins to die, the cell wall begins to disintegrate, and the lipid, the toxic lipid, is going to be released into the blood. Put that down as another problem with gram-negatives. Because if you have a gram-negative infection and you give antibiotics and you kill a lot of the gram-negatives all at once, you're going to kill the patient, because you're going to release so much of these toxins that the patient is going to die. So it's called a gram-negative intoxication. So when there is a systemic gram-negative infection, you have to be very careful how you treat it, because you don't want to have these endotoxin shock that ends up killing the patient. Typically, they either give antibiotics that slow down the growth of the bacteria without killing it and allow for the immune system to take over and clear out the infection, as opposed to just giving an antibiotic that kills everything right and left. All right, so we go back to the components of lipopolysaccharide. So lipid A is an important component. The polysaccharide that sticks out is called O-polysaccharide. It's a complex polysaccharide, which means it's antigenic, and we can use it to identify bacteria via antigen-antibody reactions. Okay. Yeah, LPS is unique to bacteria. So the presence of LPS in our bodies alerts the immune system that there is gram-negative bacteria and that it has to be eliminated. Again, lipid A is an endotoxin, which could be deadly. O polysaccharide is useful in identifying bacteria, gram-negative bacteria. So, for example, E. coli O157H7, the O in O157 is the designation for the O polysaccharide found in this gram-negative bacteria. Okay, remember we talked about foreign proteins, which are specific and selective, and they may prevent the entrance of some antibiotics. So some antibiotics cannot affect gram-negatives. For example, penicillin doesn't affect gram-negative. That's a gram-positive only antibiotic. It cannot go through the porins, so it cannot have an effect on gram-negatives. Now, what we have done with a lot of these antibiotics is we have taken them into the laboratory and we have modified them so that they can go through porins and can be used. Okay. Remember that the outer lipid layer is damaged by the short decolorization step during the gram-negative staining. And that would make the gram-negative be decolorized, and then it will take up the second dye, which is the safranin. So that's why the gram-negatives are going to end up looking red at the end of the procedure. Okay. So definitely know the difference between gram-negative and gram-positive cell walls, know what is lipid A is, know what is LPS, and we will talk about LPS and lipid A again later in the semester. In regards to other types of cell walls, there really aren't any other types of cell walls that are significantly different. We do have the bacteria that doesn't have a cell wall, which is Mycoplasma, and so that would be that lack of cell wall will make it have different characteristics. And for one thing, it doesn't have a definitive shape because there is no cell wall to keep its shape. The bacteria has different shapes. Yeah, another thing to consider is that any antibiotic that prevents the synthesis of the cell wall will only work if the bacteria is actively making cell wall. If the bacteria is dormant and is not doing anything, the antibiotic cannot work. And that is part of the problem with Mycobacterium species, is that they grow so slowly that most of these antibiotics do not work, at least do not work immediately. They may take months and months and months to work because many of them work through the chemical reaction that is making the peptidoglycan, for example. And if that reaction proceeds painfully slowly or no, it's just the antibiotic will take a lot longer to work. So that's, again, another caveat with the mycobacteria. All right, any questions about cell walls? Okay, so moving on, let's look at the other covering outside of the cell walls, which are the capsules and the slime layers. Now, those two components at one point, as a group, were called the glycocalyx. I honestly don't remember how we spell calyx. I'm gonna put it like this. This could be a Y, but I don't remember. Okay, so this is still used. This term is still used. And as a matter of fact, the virtual labs at one point do call those called capsules and slime layers glycocalyxes. Or I think it calls capsules glycocalyx. It is not technically a glycocalyx. Okay, the glycocalyx is, well, I guess you could say it is technically glycocalyx, but it's more specifically a capsule. So anything outside of the cell wall, any layer outside of the cell wall was called a glycocalyx. Turns out there are two of these layers, the capsules and the slime layers, and they're not the same thing. A capsule is highly organized, meaning that it is stable and is not going to make bonds with other molecules, which means cannot be stained. The purpose of the capsule is to protect the organism from phagocytosis of the immune system of the host. It can also be used for attachment to surfaces, and these capsules can be protein or complex polysaccharides, which means capsules could be antigenic. On the other hand, slime layers are water-soluble, easily removed. They are sticky, so they're there for attachment. And they're typically made of polysaccharides, and they're not very antigenic. There you go. It is a Y. Okay, it's a LYX. So there's glycocalyx. So we talked about capsules contributing to the microorganism's virulence by preventing phagocytosis. Bacteria that exhibit capsules, Streptococcus pneumonia, which causes pneumonia, Klebsiella pneumonia, Bacillus anthracis. And remember, we talked about the capsule stain at the beginning of this chapter as a negative stain. So in the capsule stain, the capsule is the clear area. The cell is the purple dot inside the clear area. The capsules do not stain because they're stable. So both slime layers and capsules can create biofilms. Biofilms are layers and layers and layers of bacteria sticking to each other. And they protect each other, so they are resistant to antibiotics, to disinfectants. That's Streptococcus mutans, which is a bacteria that makes slime layers, which makes the cell very sticky. And with that, they stick to the gums and create plaque. So, know the difference between slime layers and capsules. Know which one is antigenic and could be used for vaccines, for example. Which one can be detected by a negative staining. Okay, moving on. Other structures like flagella, pila, and fimbria. These are structures made of proteins that protrude out of the cell. Flagella are made by a protein called flagellin, unique to bacteria. The flagella of eukaryotes is not made of flagellin. It is used for motility. The flagella of bacteria rotate. So this is the little bacteria, and there's the flagella. The flagella begins to rotate like a corkscrew, and that makes the cell rotate. The cell exhibits is chemotactic, meaning that it could be attracted towards nutrients or away from toxins. So the cell will be swim towards a nutrient or away from a toxin. So there are proteins that detect substances at one end of the cell, and then those receptors will tell the flagella to either move towards or move away from the substances that are being detected. Flagella, because it is a protein, it is antigenic, so we could use it for classifications. So Salmonella species, for example, are classified based on their flagellar antigens. E. coli 015787. We know what 0157 is. H7 actually refers to the flagella antigen. Okay. Some bacteria have their unique flagella that wrap around their body. This is called an endoflagella or an axial filament. So spirochetes have axial filaments. Spirochetes like Treponema pallidum, which causes syphilis, and Borrelia burgdorferi, which causes Lyme disease. Now, fimbria and pila are different from flagella. Fimbria are short, sticky fibers that are there for attachment purposes. They are protein, therefore they are antigenic, and they're found mainly in gram-negatives. Example, Neisseria gonorrhea has fimbria, which uses to attach to the mucous membranes in the genitourinary tract. So in here, these long green structures are the flagella. The little red ones coming out is are the fimbria, and then other flagella can make these long protein structure that is going to be used to contact other bacteria and transfer genetic material, pieces of DNA, from one cell to another. These kinds of structures are called conjunctivae or sex pili. And we'll talk more about these on chapter 8. So, know the difference between pili and fimbria and flagella, and know the purpose of these structures. Is sex pili how strains are made? Yes, that is one of the mechanisms that create strains. Absolutely, yeah. There are 4 mechanisms that can create strains. Sex pili are one of those. Absolutely, yeah. Good catch. Internal structures. Okay, so all these structures that we have seen so far are external structures. These are layers of coverings, of coats around the cytoplasm of the bacterial cell. Now we're going to look inside the cell, and ultimately there isn't much in there because these cells do not have organelles. So the kinds of things we're going to find will be a chromosome, okay, DNA, yeah, that's there. Little pieces of DNA called plasmids, ribosomes. We'll talk a little bit about endospores. And then other things that we're not going to talk about will be things like the cytoskeleton, little granules for storage, or gas vesicles for also storage. All right, so DNA, the DNA of prokaryotes. A couple of things here. First of all, it's not inside a nucleus, that we knew. Instead, it is found in a discrete area of the cytoplasm that we're going to call the nucleoid. Most prokaryotes have one single circular double-stranded DNA molecule. So it's not like eukaryotes that have several chromosomes. In this case, there's just one chromosome. Okay, we'll talk about replication in later chapters. In contrast, eukaryotic DNA is linear, double-stranded. Okay, that's the same. And there's more than one. And obviously, it's found in the nucleus. Bacteria also have smaller pieces of DNA that are extra chromosomal, so they're not part of the chromosome. So this is the chromosome, it's a circle, and bacteria are going to have smaller pieces, circular pieces of DNA called plasmids. Our plasmids are very versatile. They replicate on their own. They can join with each other, making bigger plasmids. They can join into the DNA, creating, inserting themselves in the DNA. They carry non-essential genes, so you can remove all the plasmids from a bacteria, and that's not going to kill the bacteria. They may be able to transfer plasmids among each other, and oftentimes the genes found in plasmids give bacteria an advantage. So oftentimes the genes for toxins will be found in a plasmid. The genes for resistance to certain antibiotics are going to be found in a plasmid. So we'll talk more about plasmids later on, but for now, know that what plasmids are. Plasmids are also found in yeast, but as outside of yeast, there are no other eukaryotes that we know of right now that have plasmids. So in this picture right here, this is the cell, and this is the DNA that has spilled from the cell, and these are the plasmids of the cell. Okay. The other thing we find inside prokaryotic cells are ribosomes. Ribosomes are made of RNA and proteins, and these are the site of protein synthesis. The RNA is called ribosomal RNA, and a ribosome is made of two subunits, so two smaller pieces of ribosomes. So we have a large subunit and a smaller subunit that are going to come together. Scientists made up a unit to measure ribosomes. The unit is called the Sedberg unit, and the abbreviation is an S. So prokaryotic ribosomes have a size of 70S, and they're made of two components, a large 50S and a smaller 30S. The location of ribosomes. Ribosomes are the site of protein synthesis. So in prokaryotes, ribosomes are going to be found attached to the cell membrane and then free in the cytoplasm. So know the characteristics of prokaryotic ribosomes, know the location, the size. Contrast those with eukaryotic ribosomes, which are 80S, with two components, 60S and 40S. Eukaryotic ribosomes are found in the cytoplasm and in the inner membrane of the mitochondria. Oh, what am I saying? There too, but give me just a second. Cytoplasm and the endoplasmic reticulum. Let me take here. Next. Okay, so location where we find eukaryotic ribosomes are going to be in the cytoplasm and the endoplasmic reticulum, in which case the endoplasmic reticulum is called the ROV-R. All right, now, in the mitochondria of eukaryotes, we're going to find DNA and ribosomes. The DNA is called mitochondrial DNA. The ribosomes of the mitochondria are 70S ribosomes with a 50S and a 30S component. So they are identical to the ribosomes found in bacteria. So the theory is, this is a eukaryotic cell. The theory is that mitochondria were free living cells with their own little DNA, which were engulfed by larger cells over the process of evolution, and they became an organelle. But they have retained the characteristics of a prokaryotic cell. They have circular DNA, one piece of DNA. They have 70S ribosomes with 50S and 30S subunits. Now, why do we care about all this so much? Because of selective toxicity. Because if an antibiotic is going to bind to the 50S subunit and damage the 50S subunit of a prokaryotic of a bacteria, you would say, well, that's not going to bother our ribosomes because we don't have a 50S subunit, we have a 60S and a 40S subunit. And that's true, except the mitochondrial ribosome does have a 50S subunit. So those kinds of drugs will damage or could damage the mitochondrial ribosome. Okay, so that's the implication, is that antibiotics that damage bacterial ribosomes can potentially damage mitochondrial ribosomes. Okay. All right, any questions about ribosomes? All right, so the next thing are the endospores, which, they are structures made of peptidoglycan and proteins. They're dormant cells. They can survive for a long years and years and years in an environmental, inhospitable place, and that is dry, that has radiation, no food, extreme temperatures, and as soon as the environment improves, the spore germinates. Okay, you should know genus of bacteria that have endospores, like Bacillus and Clostridium. Okay, the process of the spore creating a living cell again is called sporulation. Sorry, I'm saying it incorrectly. Sporulation is when the cell turns into an endospore. The process in which the endospore comes back to being a live cell is germination. The cell that doesn't have spore is called a vegetative cell, the live cell. And also remember, all endospore-forming bacteria are gram-positive, but not all gram-positive are endospore-forming. Okay. All right. I think that pretty much is going to wrap it up. Make sure there's nothing else. Do endospores stop internal processes? Yes, they do. There is nothing going on inside an endospore. The endospore has a dry out DNA that has been stabilized, and everything inside the endospore is dry. There's nothing going on metabolically. It is completely dormant. And it can survive for years and years and years and years until the environment is propitious again, and then it comes back to life. So yeah, they are completely dry out environments. Okay, so as you review this material, go over, there's a chart under study guides that summarizes all of these components of the prokaryotic cell, and compare and contrast them with the components of a eukaryotic cell. So that's a good way to go back and forth. And as you're doing that, you should constantly ask yourself, oh, is this a good target for an antibiotic? Okay, so yeah, the flagella, we could get antibiotics that destroy the flagella, but that's not going to kill the cell. It's just not going to make it mobile, but that's not going to kill it. So, or the cell membrane, for example, it is so similar to a human cell membrane that it may be dangerous to use the cell membrane as a target for an antibiotic. So those are the kinds of things you need to be thinking about as you go over this chapter. Any questions? Do endospores contribute to reemergent disease virulence? No, they do not. Like if a really old endospore that's sporulated a while ago, no, they don't. That has not been known to happen. Organisms that are pathogenic that have endospores would be things like tetanus, which is very rare. I'm trying to think of another one, Bacillus tetani, botulinum, which is a food intoxication. So no, typically an endospore will not cause a reemergent. At least not that we know. It has not. Maybe the closest thing. So if there was one in King Tut's tomb, nothing would happen. Chances are, if there's one in King Tut's tomb, is the endospore of a bacteria that is probably not harmful. Now, they're saying if King Tut's died from this bacteria, that if the bacteria that is there is the one that caused his death, could it be a dangerous bacteria to humans? In that case, you could hypothesize that. Yes, you could. And let's face it, something like, did you say botulism like they use? Yes, absolutely. Yes, botulism like the Botox, absolutely. Yes, it is. Yeah. I'll come back to the botulism. The King Tut thing, closest thing we have to that scenario that you have mentioned, there were some endospores that were retrieved from vases from the Roman times, so 1,000-year-old vases, that held honey. Honey is loaded with endospores, bacterial endospores. And so there were endospores in that vase, and they were able to sporulate them. But they were harmless. They were just, Clostridium or, some harmless Clostridium or harmless Bacillus. So it was nothing that killed anything. But anthrax is Bacillus anthracis. That's an endospore. So it's been used as a bioterrorism weapon, because the endospores have been used as that. So yeah, in that regard, you could use them to kill somebody. Botulinum, yes. Botulinum, now there's a difference between the toxin and the endospore. Clostridium botulinum is an endospore former, so it does produces endospores, but it also produces a toxin, an incredibly powerful. It's one of the most powerful toxins we know of. Obviously, when you use it as Botox, you use minuscule amounts of the toxin, tiny, tiny, tiny amounts. Could it kill you if you use too much? Absolutely, it could kill you. Yes, of course. But obviously, they're not going to do that. They put tiny, tiny amounts. The toxin causes what's called flaccid paralysis. It stops the contraction of the skeletal muscles, which is nice if they're trying to get rid of wrinkles, but it could also stop the diaphragm from contracting, in which case you won't be able to breathe. So that's a different story altogether. But it's exactly the same bacteria. Torticollis too. Is it used for that? Oh, for to uh, why does it only act short term? The body will get rid of it. It's a protein, so the immune system will definitely recognize it as a foreign antigen, and it will get rid of it. Which is actually a drawback of using it as Botox, is because it is activating your immune system. So it's used for torticollis. Okay, I didn't know that. That's clever. Yeah, torticollis is when you have in the neck when your muscles are very stiff and you have a hard time moving them, and so they use Botox to relieve that constraint. Yeah. Oh well, look at you. That was the original use. I did not know that was the original use. I thought the original use was for the eyes. They also use it for the eyes for when somebody has the eye that I'm trying to think how that goes. The muscle is those children in which one of the eyes tends to move medially. Strabismus. Thank you. Yes, strabismus. There you go. That is the name. Strabismus. One eye is pointing medially, and because of the muscle being tight, so they put Botox to relax that muscle and correct it. So that might have been short and another use for it. But I did not know that torticollis was the first use for it. I thought it had been the eyes, and then along the way somebody realized that they could use it also for wrinkles, and then that's when it became famous because before that it was still being used. Um, so you had Botox and had 2 measures. Oh my goodness. Okay. So just out of yeah. So did the Botox correct it? Was the Botox after the surgeries, and that's what finally did the trick, or it was a combination of both surgeries and Botox that corrected the strabismus? Anyway, but it is definitely very potent toxins. Um, oh, I see what you're saying. Okay, I thought. Okay, got it. I thought they had used it for. Okay, hey, that's. I thought you had said. Okay, I misunderstood. All right. Yeah, they could have used it for you, but they didn't. They were not using it then. Okay, that's good. But that's just one of the uses for these kinds of toxins. We'll talk about these in chapter 16. Towards the end of the semester, we'll talk about botulinum, Clostridium botulinum. But yeah, those are definitely endospore formers. So, any other questions? All right, very good. So, I was going to start chapter 4, but I think it's a bit too much. And so let's wait chapter 4 until next week, and we'll tackle it then. Um, yes, we're going to go over lab 4 on Monday. I'm trying to think what lab 4 is. I can't remember right now. Should be control of microbial flow. What is lab 4? We just did a set of techniques. So I have to look it up. I don't remember where it is. So we have 3 labs left. Staining. Oh, it's staining. Oh, that's right. I didn't know that. Lab 4 is staining. Perfect. Okay, so lab 4 is going to cover gram stain. Yeah. Um, uh, you're having problems with Blackboard. Oh, yeah. Um, for lectures. Yeah, really. I have a room set up for labs. I thought I had a room set up for lectures. No, I do have one set up for. Oh, I know what it is. Okay. All right, listen to me, guys. This is the deal. I know what you're saying. That I know exactly what you're saying. That. All right. I have two sections here. Okay. So we're meeting here as one class when in reality we're two classes. So we have two sections, 71440 and 7120020. Okay. So this is what I did. I set up on one of the sections, I set up the room for lecture, and in the other section, I set up the room for lab. That way, kind of a split. And so those who are in the 71440 section, they have access to the recordings for lecture without waiting for me to give them the link. Those who are in the 71020 section have access to the recordings for lab without having to wait for me to give the link. So that is the problem right there. So that's why you're saying that you don't have a room for lecture, because the room for lecture is in the other section. Okay, that's the problem. So yeah, sorry guys, but that's, I had to do it this way. Okay, so what I need to do is I'm going to have to continue downloading into YouTube. And I think YouTube is working fine. I mean, I'm having absolutely no problem with YouTube. The only thing is it does take time. So it takes time, and a recording that is, let me go ahead and stop the recording here. A 1 hour